read the glycine under normal neutral amino acids and methionine under the sulfur containing amino acid. But here I want to do it combined glycine and methionine because the important compounds which are synthesized from glycine are or they require the another amino acid, that is the methionine. So that's why we will be doing this in a combined, that is glycine and methionine, two amino acids together. Okay. When it comes to the amino acid, glycine and methionine, all the amino acid per se, we are going to read it under few headings, because when in the exam they might ask you for 5-mark question most commonly, or sometimes you might expect a 15-mark nowadays pattern, which is a case-based question, or if they are following 10-mark question, then it might be having some distribution like pathway, what are the important compounds inside, etc. So both the amino acid, which is the glycine and the methionine, we are going to read it under headings, that is the general features. What is the role, biochemical function of that amino acid in the body, which includes even what are the specialized compounds formed from them, and also if there is deficiency of the enzymes in the pathway, what are the disorders we are going to encounter with. Okay, so under these headings we are going to cover these two amino acids, that is the glycine and the methionine. Okay, so first coming to the first amino acid, glycine. When we see the glycine amino acid and the classification of amino acid also, it is the most simple. Why we call it simple? Because when you see the structure, two functional groups of the amino acid, R group is replaced by a simple H and a H. Okay, so there is no large R group, it is just simple H. So it's a small simple amino acid, and both the functional groups are attached to the first carbon. That's why these all naturally occurring amino acids, we call them as the alpha amino acids. And second feature of this amino acid is it is non-essential. Okay, if you remind yourself for the classification of the amino acid based on the requirement of the diet, we classify them as either essential amino acids or the non-essential amino acids. Essential means you need to take in your diet on daily basis. Non-essential means you need not to take it on a regular basis for the diet. Okay, means there is a mechanism by which these can be synthesized in the body or converted from other amino acids. So that's why we call them as the non-essential, because they can be synthesized within our own body. Next, fate of the amino acid. In again a classification of amino acids, we have something called as glucogenic amino acids, means they help, or the carbon particle or the skeleton of the amino acid helps in glucose synthesis, or ketogenic if they help in the synthesis of ketone bodies, or some are combined, partly glucogenic and partly ketogenic. When it comes to glycine, it is glucogenic in nature. Okay, the carbon particles or the skeleton of the glycine is used to synthesize glucose in the body. Again, when you go back, the gluconeogenesis occurs only in the liver. The glucose synthesis of from any amino acid or any non-carbohydrate molecules is always in the liver. The glucogenesis only occurs in the liver. The liver can contribute glucose to the bloodstream. So this is the general feature of glycine. Now, few things we have to know in glycine. One is how it can be synthesized, or what are the sources of glycine. Okay, when we see the source of glycine, we have some sources like, for example, amino acid serine. Serine can be converted into glycine. Okay, serine can be converted into glycine. Second is one more compound, threonine. Okay, threonine also can synthesize the amino acid glycine. Okay, next, this can be synthesized from scratch. Okay, that is by a synthase complex. Okay, by a synthase complex, because it is synthesizing glycine, it is called as glycine synthase complex. So by this complex also we can generate the glycine. Now second, this is about the source. Next, what is the degradation? Okay, if you degrade the glycine, means you dismantle the glycine, what you are going to get? We all know glycine is made up of carboxyl group, amino group, carbon, and 2 hydrogens. So when you dismantle it, definitely we are going to get these compounds individually, separated from each other. Okay, so this is called as glycine degradation system. Okay, glycine degradation system, or also called as glycine cleavage system. Again, in the exam, this is a short answer question. Okay, that is a 3-mark question in the exam. That is a glycine cleavage system. So, glycine cleavage system is a multi-enzyme complex. Means it is not made up of a single enzyme, but it is containing the multiple enzymes. So, it's a multi-enzyme complex which helps in the degradation of the glycine. Okay, so when it comes to this system of glycine degradation, the glycine degradation requires one important compound. Again, pyridoxal phosphate. We have seen in transamination reaction. Transamination reaction also requires the PLP. When you see the glycine breaking down into small molecules, which is the ammonia, carbon dioxide. So again, we require the pyridoxal phosphate or PLP. Okay. So when you see the reaction, glycine is converted into a compound amino methyl group. So if you see here, what we are removing? We are removing carbon dioxide. The carboxyl group COO is removed as carbon dioxide. Now this further will be released as ammonia. Now this is also removed. Now what is left behind? C, 1H, 2H and 3H. What is left behind is methyl group. We know that there is something called as tetrahydrofolic acid, who are called as 1 carbon carriers. So they will help in carrying the methyl group. What is left behind? They will carry them. Okay, methyl group is taken away by the THF, tetrahydrofolic acid. That is the active form of folic acid. What is folic acid again? It's a vitamin. Specifically, it's a water-soluble vitamin, which is important in metabolism of amino acids. Again, I'll stress on this thing. Most of the water-soluble vitamins, they act as coenzymes and carriers of the carbon particles in the metabolism of amino acids. So if there is deficiency of some of the amino acids and vitamins, definitely they will impact the metabolism of these things. Like for example, if PLP is absent, pyridoxal phosphate, it is again a water-soluble vitamin active form. So this is absent, glycine to amino methyl group is hampered. Further, if folic acid is absent, then the release of ammonia is also hampered. Okay, so that is how the vitamins, that specifically water-soluble vitamins, play a major role in metabolism here with respect to the amino acids of the protein metabolism. Okay, so this is called as glycine cleavage system. This is an important 3-mark question in the exam, you might expect. Now, first enzyme, we are removing the carbon dioxide. So, first enzyme will be decarboxylase. Because we are removing the carbon dioxide, then it is called as decarboxylase enzyme. Whom we are decarboxylating? Glycine. So, it will be glycine decarboxylase enzyme. Okay, that is the first enzyme. Next, second enzyme. What is the second enzyme? What we are doing? We are removing the ammonia. We are transferring the methyl group to the THFA to form methylene THFA. We are transferring the methyl group. So what will be the enzyme? That is the transferase. What we are transferring? The
methyl group.
Yes. From amino methyl. So it's a amino methyl transferase enzyme. So these are the 2 important enzymes which are very much mandatory when you are writing the degradation. It's a very simple. What we are removing? First, from carboxyl group, we are removing carbon dioxide, CO2. Decarboxylase enzyme will take care of that. Next, we are removing the amino group and also whatever left behind CH3. What is left behind for us is amino methyl group. Amino methyl group, from that we are transferring the methyl group, 1 carbon carriers. 1 carbon carriers are the THFA, tetrahydrofolic acid. It's a folic acid active form. So what it will do, it will come into the reaction, takes away the methyl group and goes away with methyl tetrahydrofolate. So it is the amino methyl transferase enzyme. Again, what is left behind is ammonia. What will happen to this ammonia? Yesterday we have seen different fates are, different fates are there. In that, majorly it will undergo the urea cycle. Okay, it will undergo the urea cycle. That is again in liver only. Okay, major urea cycle occurs in the liver. So this is about degradation. So we have seen what is the source of glycine. We have seen how it is degraded. Now, next important question in the exam comes is the what are the like functions or clinical application of this. Okay, before that, what we'll read is the what are the what you call the important compounds or specialized compounds. Okay, specialized compounds synthesized. Okay, specialized compound synthesis. It's again a 3-mark question in exam or 5-mark question, which can be a short answer or short essay, which our university might be. But glycine specialized compound synthesis itself is the important question in the exam. Okay, please remember. So when it comes to specialized compound synthesis from the glycine, we have different compounds. To summarize it, if you see glycine, one side we can synthesize important compounds. Okay, important compounds. On other side, there is source. Source can be from diet. Source can be from serine amino acid. Source can be from threonine. Source can be from synthase enzyme complex. Okay, fresh synthesis. So, and catabolism of a already existing protein. So if you catabolize the protein, again you will get small amino acids. In that you are going to get your glycine as well. What are the important compounds itself from glycine? Again, glycine can undergo conversion into serine. It's a reversible reaction. Serine can give to glycine, glycine can give rise to serine. Okay, next important compounds includes glyoxylic acid or oxalates. Okay, that is synthesized. Next, most important, creatine. That is creatine phosphate, what you see in the muscles. The creatine part is synthesized from the glycine. This itself is an important 5-mark question in the exam or 3-mark question in the exam. Okay, next, it can contribute for purine ring. Okay, it can contribute for the purine ring. Specifically, which carbons? C4, 5, and 7. Okay, 4, 5, and 7 are the carbons which are contributed from the glycine structure itself. The glycine will contribute to the 4, 5, and 7. In that, 4, 5 is carbon and 7 is nitrogen. Okay, glycine is contributing. Next, if you go back to the hemoglobin, heme is synthesized with help of the glycine. So if there is deficiency of glycine, hemoglobin synthesis also will be hampered. And yesterday we have seen one more compound called as glutathione. What is glutathione? It's a gamma glutamyl cysteine glycine. So glycine is the important part of glutathione. Okay, what are the roles of glutathione? What is glutathione? That will be the part of chemistry of amino acids. In that you come across something called as biological important peptides. So in that, glutathione is a tripeptide. Okay, it contains 3 amino acids, gamma, because it is in the gamma position, gamma glutamyl cysteine glycine. So glutathione synthesis. And when you are reading degradation of hemoglobin and derivatives of the cholesterol, we come across something called as bile salts. So bile salts are again synthesized with conjugation with glycine and taurine. So glycine itself is important part of the bile salt synthesis. And last one, it helps in conjugation. Okay, why the body does conjugation is they make the compounds more water soluble. Okay, in the body, some of the compounds are toxic. They undergo something called as detoxification process. In that, one of the mechanism is either you add the hydroxy group, OH group to the molecule so that it becomes water soluble and excreted through urine, or it can be conjugated. So one of the example is hippuric acid. Okay, example is hippuric acid. So these are the important compounds synthesized from glycine. It's an important 3-mark question in exam. What are the compounds? At least you should be able to write 4 to 6 compounds which are synthesized from glycine. Is it remember creatine, creatine phosphate, glyoxylic acid, which is nothing but phenyl oxalate. Then it helps in synthesis of purine. Specifically, if you don't remember which carbon, which nitrogen, that's also fine. But glycine helps in the synthesis of purine. Hemoglobin, the heme part of the hemoglobin, the first step itself requires the amino acid glycine. Then glutathione molecule, which is tripeptide. By nature, it contains the glycine in the structure. Apart from these things, bile salts and conjugation process in the detoxification conjugation process requires the glycine. So these are the compounds synthesized from or specialized compounds from glycine. Okay. So I hope this is clear. Yes.
So you're not audible.
Is it not audible? Hello.
So now it is audible. It would one it would be a kind of close.
Okay. Till end, did you follow?
Yes, sir. So mean that hydrochloric acid.
Okay. Yeah. So this is a very important 3-mark question in the exam. They will ask you to write about specialized compound synthesized from glycine. In that, at least you should be able to write 4 to 6 important compounds synthesized. Okay. And sometimes if they are asking for 5-mark question, what they'll ask is, one, they'll ask you to write specialized compounds synthesized from glycine and note on creatine synthesis. Okay. Note on creatine synthesis. This itself is an important question. This can be a 3-mark question or a 5-mark question. That is this portion. Okay. So now we'll see creatine synthesis. Okay. Till now we have seen what are the important specialized compounds from the glycine. Now we'll follow the creatine synthesis. Okay. Is it clear now?
Yes.
Okay. Yes. Very good. Sure. So we'll go about creatine. Now please don't confuse yourself from creatinine and creatine. Okay. Creatine is the compound synthesized from the glycine. Creatinine is the waste compound which is synthesized from the same molecule. Okay. Creatine, after its half-life, after its life, it is converted into a waste compound. That is called as creatinine. Okay. Creatinine is a waste compound. Creatine is a biological compound synthesized from glycine. In the muscles, you might have followed in physiology, we have something called as creatine phosphate. Okay, so creatine phosphate is nothing but creatine binding with a phosphate group. From where is this phosphate coming? Always remember, the phosphate is always donated from majorly the ATP. Sometimes some reactions GTP come into picture, sometimes CTP, but majorly it is through the ATP. Okay, adenosine triphosphate. Okay, now next we'll see how this is synthesized, which is a 5-mark question. How this is synthesized is the creatine. Now, what is the main substrate required? Substrate is same. We require glycine. Without glycine, we cannot synthesize the creatine. Second important compound is the methionine. But methionine will not take part as methionine. It will take part as its active form. Okay, active form of methionine. That is called as S-adenosyl methionine. Means what is added? Adenosyl group. Adenosyl, from where it comes again? ATP, adenosyl triphosphate. So adenosyl group comes from the ATP. What is the active form of methionine? It's also called as, yes, it is also called as SAM. Okay, SAM means S-adenosyl methionine. And next, one more amino acid is important in this reaction. That is the arginine. Okay, so these are the 3 important substrates what are required for the reaction, major substrates. Second, what is the end product? End product is what we are synthesizing. Creatine, which is further converted into creatine phosphate. So that is our end product. In between, what we might require, because we require the phosphate groups, we require important compounds. Here are ATP. Okay, so these are the 3 things which are very important in the reaction. That is the glycine conversion into creatine and creatine into creatine phosphate. 3 major amino acids play an important role: glycine, methionine, and arginine. Methionine will not take part as methionine, but through its active form, that is called as S-adenosyl methionine, or also called as SAM. So this is a general what we are going to see. Now we'll see the steps. Steps of the creatine synthesis. Okay, when it comes to steps, first we'll write our main important compound, substrate, glycine. It reacts with amido group containing amino acid, that is arginine. Okay, arginine. Glycine reacting with arginine to synthesize a compound. Yesterday we have seen one compound, that is ornithine. And glycine is converted with the reacting or condensation with the arginine to synthesize an important compound, guanidine. Okay, guanido acetic acid. As I told you, arginine is a important amino acid which contains guanidino group. Okay, the specialized what you call the molecule which is present in the arginine is guanidino group. Like methionine contains important compound is sulfur, sulfur containing amino acid. Serine, hydroxy containing amino acid. Similarly, arginine is guanidino group containing amino acid like tyrosine, benzene ring, proline, imino group. Yes. Similarly, arginine is a guanidino group containing. So glycine condensing with arginine to synthesize guanidinoacetic acid and ornithine. Next, what happens to this guanidinoacetic acid? It is converted into creatine. Always follow the same. First, we always try to memorize the basic backbone. Okay, the basic background of the reaction. Don't directly jump into the reaction and the enzyme because it's very difficult to remember two things at the same time. First, we will learn the backbone. Then we'll add the enzymes into the reactions. Okay, is it clear?
Yes, yes.
Because that is the simplest way how we can remember the pathways. If you start reading A converted to B with enzyme E1 is difficult. Rather than we'll complete whole reactions from starting to ending so that we don't miss the flow. And then we start inserting the enzymes who is helping the reaction. So first, glycine reacting with arginine, guanidinoacetic acid group is synthesized. Ornithine is released from the reaction. Guanidinoacetic acid is converted into creatine, and further creatine is converted into creatine phosphate. So what was our steps? We wanted to synthesize glycine to creatine. So we got from glycine to creatine and then creatine phosphate. Now when we come to the first enzyme, what we are transferring, amido group is transferred. So it is a glycine, arginine, amido transferase. Yes, clear? Glycine, arginine. Arginine, what it is doing? It is donating the amido group to glycine. So it is a transferase enzyme. It's a glycine, arginine, amido transferase. In the exam, another simple trick. If you don't remember the whole name, if you are able to even locate what enzyme class it belongs to, that also is sufficient to score the marks. Okay, sometimes we don't remember. Sometimes the enzyme name will be based on the product, guanidinoacetic acid transferase. Sometimes it will be the substrate, glycine, arginine, amido transferase. If we don't remember such things, then always try to remember at least the class. Like in previous reactions we have seen, carbon dioxide is removed. So what class of enzyme? Decarboxylase. We are removing, yes, we are removing carbon dioxide. So that's why if you don't remember what is the first name of the enzyme, try to remember the class of enzyme. Okay, what class the enzyme belongs to? Is it transferring? If it is synthesizing something, synthase. If synthesis requires ATP, then we add the T in the synthase. It becomes synthetase. Okay, so that is the simplest method how you can remember the enzymes. Now this guanidinoacetic acid is converted into creatine by accepting methyl group. Now we have seen there is one more compound required. Yes, adenosyl methionine. Okay, if you remember the chemistries of amino acids, in that there are some general reactions, transmethylation reactions. Okay, the transmethylation reactions. The transmethylation reactions are primarily dependent on one amino acid, that is the methionine. That too specifically, if I go the S-adenosyl methionine, that is the SAM. So this SAM comes into picture here. Yes, adenosyl methionine donates the methyl group to the guanidinoacetic acid to synthesize creatine, and from SAH it becomes SAM to SAH. That is the homocysteine. Okay, yes, adenosyl homocysteine. Yes, adenosyl methionine is converted into S-adenosyl homocysteine. Again, what it is happening? Transferring. So it's a class of transferase enzyme. What it is transferring? Substrate guanido. Yes, it is the enzyme guanido acetate. Okay, it is transferring methyl as you told. Very good. Methyl transferase. Substrate is guanidinoacetic acid. Acetic acid we can write as acetate also. Okay, acetic acid or acetate. Guanido acetate methyl transferase. Methyl group is transferred from SAM, S-adenosyl methionine, to guanidinoacetic acid to synthesize our important compound what we required. Is it stopping here? No, we want phosphate group to be added. Who is adding the phosphate? ATP. Yes, ATP is converted into ADP plus phosphate is added. Most commonly, these reactions are done by a class of enzyme kinases. Okay, so kinases helps in the picture. Substrate is creatine. So it's a creatine kinase. So creatine kinase converts creatine into creatine phosphate. Now this will act on the muscles. It acts as a small packets of energy like cells. Definitely the major fuel or energy molecule in the muscle is the ATP. But the sudden gush which is required on a muscle contraction, that short cells is the creatine phosphate, the phosphate group. Because once ATP are utilized, they are exhausted. Till the ATP are generated by the mechanism. We have mechanism like we have glycolysis, we have PCA cycle, we have electron transport chain, we have beta oxidation. So all these major machineries will take some time to kickstart. But you cannot stop the muscle from acting. So that short backup, like for example, you have got a computer, electricity is gone, generator will take some 10 seconds of time. But still you don't want your computer to switch off. What we have UPS backup. So similarly, muscle has UPS backup, which is called as creatine phosphate. What creatine phosphate will do? It will donate the phosphate group to ADP. ADP will convert into ATP. It will help in the muscle contraction. So that is the reason why we have to synthesize this short molecules, which we call as the battery backup for the ATP synthesis in the muscle specifically, is the creatine phosphate. Now, once creatine phosphate synthesized, is it for lifetime? No. It has got its own life. It will die. Once the wear and tear, once the function of the creatine is completed, it will be removed from the body. How it is removed? We are going to remove it as I told you already. The waste product from the creatine is called as creatinine. Yes. Please don't get confused between creatinine and creatine. Creatine is the specialized compound which has designed function. When it comes to creatinine, it's a waste compound which is end product of the creatine phosphate. What we are removing? We are removing the phosphate group and also we are dehydrating it. We are removing the water from the creatine to form a molecule creatinine. Now, what is the enzyme? There is no enzyme. Okay, it's a non-enzymatic reaction. It is a non-enzymatic reaction where the creatine phosphate, the creatine phosphate is undergoing dehydration. Okay, it is undergoing dehydration and removal of the phosphate group. Okay, if I show you clearly on the another picture, we have creatine phosphate. So once the lifespan is done, it is converted into creatinine, which is a waste compound with removal of PI, the phosphate group, and also dehydration, that is the water molecule. Enzyme, no enzyme. Okay, it's a non-enzymatic reaction. Once the lifespan of creatine phosphate is done, it is excreted or the waste compound is creatinine. Now, if someone asks you, is there any storage of this creatinine? What will be your answer? No. Why?
No.
Because body will not store anything which is waste. Okay. Unless it has got some design function, body will store. If it is a waste, there is no storage form. What is the location of creatine phosphate most commonly? It is the muscles.
Muscle.
Yes, it is the muscle. It can be smooth muscle, it can be a striated muscles, whatever muscle it is the muscle where is the location, creatine phosphate. The end result, end product of waste compounds synthesized from creatine phosphate is creatinine. Now, next questions come is it constantly synthesized? Yes. The muscle protein or the creatine is constantly broken down into creatinine, and creatinine is water soluble. So if it is a water soluble, what is the route of excretion? Most commonly kidney. Yes, urine, through urine formation. Okay, in the urine it is excreted. So yesterday we have seen one more compound, that is the urea. Okay, urea is also major excretory route in the urine unless urea is synthesized in the GI tract. In the GI tract, we have got bacteria. So bacteria can also utilize the ammonia and convert into urea. So that's how urea can be excreted in the feces. But this is very minor because bacteria are less in number. When it comes to the body, endogenously synthesized, it is through the urine. Okay, the kidney will release the urea and also it will excrete the creatinine. Because creatinine is constantly excreted in the urine, you can use this as a marker of kidney disease. Yesterday also I told you, urea and creatinine both can become a good marker for renal function. If kidney is not functioning properly, then blood level of these compounds will start increasing because it has to be excreted in kidney, excreted through kidney, but it is not happening. How about synthesis? Synthesis is constantly happening. Synthesis is there, output is zero. So what is happening? It is started accumulating in the blood. So these are the dictators or they are the indicators of renal function. Kidney is normal, blood levels are normal. Kidney function is low, the blood level starts increasing. And what about urine excretion? It starts reducing because it is not excreted in urine. The urine concentration of both creatinine and urea decreases. Because it is not excreted, blood level starts increasing. Synthesis is constant. It is happening continuously. Urea is also synthesized. Creatinine is also synthesized. Okay. So normal blood level of creatinine, we take it on general less than 1.0 milligram per dl or deciliter. Okay, that is the creatinine. Anything more than 1 milligram per deciliter is indicating that there is some problem with the kidney. Okay, the renal function is hampered or renal function is affected. In such cases, you will start increasing. You can see the increase in the creatinine. It can be 1, 2, 3, 4, and he might go up to 10, 15 also if there is complete renal failure or kidney shutdown. Okay, so this is about the creatine and then creatine phosphate and then excretory compound creatinine. Okay, so if we go back and see, the creatine is synthesized from glycine. It's an important 5-mark question. Glycine condenses with arginine to synthesize guanidinoacetic acid and ornithine with the help of enzyme transferase. Guanidinoacetic acid is converted into creatine with the help of another amino acid, S-adenosylmethionine. Creatine is converted into its active form, that is creatine phosphate, by accepting the ATP, that is the phosphate group, by the enzyme kinases. It will do its function, deliver its number of days, and then it is excreted. The excretory form of creatine is creatine phosphate. Because it's a waste, there is no storage form of creatinine. Because synthesis is constant, being a water soluble excreted in urine, so we can use creatinine as an important marker for kidney function. Okay, it's an important marker for kidney function. If any change in creatinine means there is change in kidney also. Kidney function is also affected. Kidney is not functioning, creatinine is very high. Kidney is normal functioning, creatinine in the normal range. To tell whether it is normal or abnormal, what we should know? We should know normal level first. Simple creatinine normal value, easy to remember, universally accepted is 1. Some books gives even 1.2, 1.4, but ideally and universally accepted is 1. Even some cases you can see 0.8 also. But to remember for the exam point of view and as an MBBS student, we should not make mistake that 1.2, 1.4 is normal. No. 1 or less than 1. Okay, less than 1 milligram per deciliter is the normal range of creatinine in blood. Okay, creatinine in the blood. So this is about the creatine phosphate. Okay, next we will learn about the what you call the metabolic errors. Okay, the last section is the inborn errors. Okay, inborn errors of metabolism. Just give a second. Okay, so inborn errors of glycine is not so importantly asked, but still you can expect a 3-mark question out of this topic. There is something called as 1 non-ketotic hyperglycinemia, not hyperglycemia. It's a glycine, hyperglycinemia. Second, primary hyperoxaluria. I told you there is one compound synthesized from glycine. That is called as oxalates. Glyoxalates. Okay, so that is related to the oxalate synthesis. So when in the exam they'll ask you, write disorder's name. What are the inborn errors associated with the glycine? 2 major important. One is called as non-ketotic hyperglycinemia, not glycemia. Glycemia means sugar. Hyperglycemia means increase in sugar levels. Glycinemia means it is related to the glycine. Okay, hyperglycinemia. It's also abbreviated as NKH. Okay, it is abbreviated as non-ketotic hyperglycinemia. It is an important question in the exam. So we'll see just what is required for exam point of view. NKH. When it comes to NKH, I told you inborn errors are all genetic disorders. Okay. What is the defect? It is defect in glycine cleavage system. What is glycine's cleavage system? We are going to completely dismantle the glycine into three compounds: methyl group, carbon dioxide, and ammonia. We are completely dismantling the glycine into these three compounds. If the cleavage system is absent, you cannot synthesize this. There is no degradation of the glycine. The glycine starts getting accumulated in the body. So what happens if you do investigations like, for example, CSF you are analyzing? Glycine concentration is very high. Okay, increase in glycine levels in the CSF. CSF is derived from what? Plasma. Again, when CSF is high, it is derived from plasma. Definitely plasma concentration also is high. Okay, and there is also most commonly affected organ will be the brain, kidney, and liver. Liver is the major metabolism. So you can see these compounds. Because brain is affected, definitely the person will start having the mental derangement or mental retardation. When brain is damaged, neurons are damaged, definitely he'll start having the regression. His skills are regressed. Initially, he was able to, like, say about brush his teeth. Now he is not even able to hold the brush because he is regressing. Normally, it is growing. A child from 1 year to 2 years, he'll develop the skills. Now it is reverse, 2 to 1 year. Okay, so you can see that part. Then muscles become hypotonic. Hypotonia can be seen because neuronal tissue. We have seen glycine is required to synthesize neurotransmitter. Okay, so if that is hampered, the person end up having the seizures. Okay, which we also called as fits in layman's term. So seizures can be seen if it is in very young newborn. So we might see some brain malformations. Okay, brain malformations can also be appreciated in the children because inborn errors are seen in the child from the birth. So you can see growth of the child decreased. Feeding, because it has to feed on the mother's breast. Feeding is decreased because hypotonia, decreased muscle strength. So it cannot suck the breast milk also. So feeding is poor. And sometimes even spasticity can be seen in the muscles. So these are the things can be seen in these cases. So if you investigate further, you do MRI scan, you end up seeing some atrophy of the brain tissue. Okay. How do you diagnose? You can diagnose. Okay. You can diagnose by simple measure the glycine levels. What you'll see, increase in glycine. Because it is water soluble, urine is also high. Urea. Okay. Glycinuria. Hyperglycinuria. Because it's water soluble. In the blood also it is increased. In the urine also it is increased. So you can diagnose by these things. One feature of this is there is ammonia levels are normal. Acidosis not present. Ketone bodies normal range. What is increased is only glycine in CSF, blood, and urine. Yes. So that's why this disorder we call as non-ketotic, because ketone bodies or acidosis is not seen. Non-ketotic. What is increased? Glycine. So it is called as hyperglycinemia. So that's why it's called as NKH. Okay. NKH is the disorder. Now next disorder is called as second oxalates, related to oxalates.
Hyperoxaluria.
Oxaluria, yes. It is related to oxalates. It is excreted in urine. Why it is excreted in urine? Because the concentration is increased. So that's why this disorder is called as hyperoxaluria. Increased in the oxalate levels which are excreted in the urine. That's why this is called as hyperoxaluria, which is seen in these cases. Okay. Normally what happens is glycine is converted into glyoxylic acid, which is converted into glyoxylates, which is converted into oxalates. Now, because it is not undergoing glycine cleavage system because of some disorder, it starts synthesizing high concentration of these oxalates and glyoxylates, which are water soluble. Second most important feature of this is they form crystals. Because they're water soluble, they form crystals. Water soluble compound means it is excreted in kidney. Because they can form crystals, the person end up having most of commonly kidney stones or renal stones. What is this stone made up of? Primarily oxalates or glyoxalates. Okay, so this is the main what you call the key feature of this disorder, that is the hyperoxaluria, where you are seeing the oxalate crystals excreted in the urine. Because oxalates are water soluble, because their concentration is very high, because lot of glycine is converted into these compounds, very high concentration, because they're excreted in urine. If, for example, person is suffering with this disorder and he takes less amount of water, so what happens? There is no flushing of the tubules. There is no flushing of the renal tubules. Nephrons are also blocked because of the formation of crystals. Normally, if the person drinks a lot of water, the small crystals get excreted or eliminated in the urine. Because if person is staying in, like say, about high temperature areas, okay, or water scarcity area, he's a labor, and he's also having this disorder, then he has the higher chance of repeated renal stones. Like person has went to a doctor with renal stone, that is in say about in 1 year 10 times. It's a huge number. What if the person goes second time? They should be alarming. What is this causing the repeated formation of the renal stones? What you will do? You collect these stones which are removed from the kidney, send it for analysis, and see what it is made up of. Is it calcium? Is it phosphorus? Or is it something like this oxalates? If it is oxalate, measure what is the glycine levels in the blood. You will see glycine levels are very high. Then you can correlate the high glycine, oxalate stones are repeated. So this person might be having the hyperoxaluria disorder. What you'll suggest to the patient? Definitely low glycine diet, because glycine by nature is a non-essential. Okay, if you don't give in food also, body can synthesize the glycine. What is the difference? If you take or take in the diet, glycine is in very high concentration. If body synthesizes glycine, definitely it will be synthesized to the extent which is required for the body. So glycine can be utilized for all its specialized compounds rather than synthesizing oxalates. Okay, so low glycine diet. Second, increased water intake, because if at all oxalates are formed, is excreted in the urine. Second, that is third, is decrease green leafy vegetables, because leafy vegetables by nature contains oxalates. Okay, they're rich in oxalates, green leafy vegetables. From glycine also oxalates are increasing. Green leafy vegetables also oxalates are coming. So the person end up having the oxalate stones or renal stones because in this disorder. Okay, is it clear with this? Yes.
Yes, sir.
Yes.
Yes.
Okay, very clear. So with this, we conclude on glycine. Okay, so glycine itself can be a 15-mark question. That is long essay, which is most commonly asked as a case-based question. Glycine can be a 10-mark question if pattern is there. It can be divided under different headings. It can be a 5-mark question, simple creatine synthesis. It can be 3-mark question, like specialized compounds synthesized. Can be a 3-mark question. Creatine phosphate synthesis itself sometimes can be a 3-mark question. Importance of creatinine can be a 3-mark question. Okay, so these are the things. And whenever you are reading, you should always read some introduction of the same glycine. What are the sources? What are the what you call the fate? What are the specialized compounds synthesized? And what are the disorders related to the same for the glycine?